Okay, we're going to just continue with macromolecules and look at proteins. So again, it's just looking at the structure and how that affects the function of the protein. Proteins are one of the most complex and diverse classes of biomolecules. They're also considered one of the most important. They're one of the most studied, definitely. Last week we talked about nucleic acids. One of the problems with the work involving nucleic acids was that most people thought that they were too simple to encode for the genetic information that we now know they do code for, and a lot of people thought it was in fact proteins because they had a lot more complexity, and they thought that more complexity meant more versatility to code for genetic information. We know that's not true now, but you can kind of see why when we start looking at proteins. They're incredibly complex molecules. So a protein consists of one or more chains of individual amino acids. The chains are called polypeptides, and they're called that because the amino acids are bonded together in peptide bonds. So polypeptide, multiple peptide bonds in a chain. So first, what we're going to do, we're going to have a look at the basic structure of an amino acid. So like the generic structure, and it's right here. So an amino acid is fairly straightforward. It's a small molecule consisting of two carbons and a nitrogen on its main chain. So it's got like a main chain segment here, and then it's got a variable side chain down here. Now this is an amine group, so thus the amino part of the name, amine, and this is a carboxylic acid, which we've seen before, and so thus the acid part of the name. So it's a carbon with an amine group and a carboxylic acid group, so it's called an amino acid. This R group, R just means a variable group. There are 20 main amino acids that occur during protein synthesis. There are other weird ones, but 20 ones you usually encounter, and each of them have a different variable R group, and the chemistry of that amino acid and its chemistry and interactions within a protein are mostly dependent on the R group. So when the R group is hydrophobic, you generally find it those sort of side chains tucked inside the protein, within the protein, away from the aqueous environment outside the protein in the cytosol or something. Whereas ionic side chains, the charged side chains, are generally found either at the interface with the aqueous environment or bonded to another charged side chain, making a ionic bridge. So different side chains are crucial to the structure, interaction of the structure of the protein, interaction of the protein with the outside environment, and the catalytic activity or binding activity of that protein. So we'll have a look at what happens with peptide bond formation. So when you have a peptide bond, you have two amino acids. So amino acid 1 here and amino acid 2 here, and we have each of them have different side chain, which I've just called R1 and R2 to keep it simple. And so what happens is the carboxylic acid group reacts with the amine group on the other amino acid, and you have this condensation reaction. So this OH group here and this H group, they sort of condense out as water, so they come out as water, and you have a bond made between this carbon and this nitrogen, and that bond here formed because they each lose a group off them, and they form a new bond through that. This group is called a peptide bond, and it bonds these two amino acids together, and you get, this is now a dipeptide, and if we kept adding, we get a polypeptide, which is what you're looking for a protein. So polypeptides generally contain like hundreds or thousands of amino acids, and so you have many different side chains, and they're generally organized into different domains, and so you have side chains arranged in a way that allows them to undertake their function and keep the structure of the protein right. The structure of proteins is generally very interlinked with its function, so it needs to be the right shape and have the right amino acid residues in the right places to be able to undertake its function, whether that might be as an enzyme or as a structural protein or something. And as we said, a protein is one or more polypeptides plus other groups. So proteins can have a lot of other prosthetic groups. For example, hemoglobin has the prosthetic group heme, which contains iron, and something like, I think it's a quarter of all proteins or a third of all proteins actually have like inorganic groups attached to them that are crucial in their activity biologically. So it is proteins aren't just amino acids. There's a lot of other groups that get added. They can undergo a bunch of other modifications, like glycosylation, where you add a small chain of a small polysaccharide chain or disaccharide group, or you can do a bunch of other modifications. But the main backbone of a protein is the amino acid backbone, and proteins do various things within a cell. So some of the functions they undertake, so we have them as enzymes. So they act as biological catalysts, and we'll look at that further in I think in a couple of lectures. When we look at kinetics, we look strongly at how enzymes work, but essentially they just speed up reactions. So biological reactions need to go fairly fast usually to keep the metabolism of the cell going, but usually to make a reaction go faster, you'd heat it, and that doesn't always work for biological cells. So you use an enzyme or a biological catalyst to speed it up. Proteins can also act as hormones. So like we have insulin, which is involved in diabetes. It is a hormone involved in sugar balance in the blood. We have carrier molecules, so like I mentioned, hemoglobin, which carries oxygen around the body. That's crucial. And then we have structural molecules like keratin. Keratin's a protein that makes up pretty much all of your hair, so and nails. So that's a fairly important structural protein as well. So that was just a brief rundown on some of the basic structure and function of proteins. Further lectures will look at how enzymes work, specifically how they speed up reactions, and we'll start to look at the different levels of structure in proteins. Proteins are a very crucial type of macromolecule in our body, and we'll see them come up a lot throughout the rest of these videos.